Okay, let's unpack this. We are about to do a deep dive into something that, honestly, sounds a bit like science fiction.
Right.
We're talking about a quest to give the human body its own built-in tracking system, a living, readable light show that can tell doctors exactly where a disease is.
A tiny cluster of cancer cells, for instance, or a hidden infection.
Exactly. And our sources today, they detail the tools we have now, but also, and this is the fascinating part, the really surprising places scientists are looking for that next big breakthrough.
It's the holy grail of non-invasive diagnosis, really. Imagine being able to track cells and how a disease is progressing deep inside the body.
For months, years.
Potentially, yes.
Right.
And all non-invasively. No high doses of radiation, no repeated painful biopsies. It gets rid of those diagnostic blind spots we're always struggling with.
And the mission for this deep dive is to really get into the physics and biology of it, to look at bioluminescence, at fluorescence, and figure out how we get past the hurdles of our current tools so we can finally see what's happening deep inside us.
Using safe, readable light.
So, to start, we have to talk about the current champion in the lab, the tool everyone uses right now. It's an enzyme, a luciferase called nanoluke.
Ah, yes.
Nanoluke. It's synthetic. It's engineered to be incredibly bright, incredibly fast. In a petri dish, it is fantastic.
It's the workhorse, for sure. But, its performance in the lab is exactly what shows us its biggest limitations for, say, whole-body human tracking.
Right.
If we want to get out of the lab, beyond just mouse models, we have to admit that nanoluke fails on three key requirements for any kind of long-term human diagnostic.
Okay, so let's take the first one. Sounds like a simple logistics issue. Okay. Fuel. Why can't we just keep feeding the cells we want to track?
Because nanoluke, like any true bioluminescent system, it needs a chemical food source, a substrate.
Yeah. Right.
For nanoluke, that's a specific compound called furamazine. The reaction is simple. Luciferase plus furamazine, you get light. So no furamazine.
No glow.
Yeah.
Which brings up the big problem of getting it there in the first place.
Precisely.
Yeah.
To get any kind of consistent, measurable light, you need a really high concentration of furamazine, and it has to reach every single cell you're targeting.
Even if that cell is, I don't know, deep inside a lung or somewhere hard to reach?
Deep in a lung, hidden in a bone marrow pocket, you name it. Delivering a stable amount of an external chemical through the body's complex barrier systems, especially for a long time, it's functionally impossible.
The body just breaks it down too fast.
It metabolizes it incredibly quickly. And trying to maintain that dose would likely come with some pretty severe toxicity risks.
So, okay, even if you could deliver the fuel safely, the cells only glow as long as they're eating. Which I guess brings us to the third problem, longevity.
Yes. Even in a perfect scenario, once that external fuel runs out and in the body, that happens fast, the glow just stops within minutes.
You get a snapshot, not a future film.
Exactly. You can't track a metastatic cancer cell over months or years, which is what you need for a real diagnostic tool.
But for me, the biggest deal breaker, the one that's pure physics, is the color of the light itself. Nanoluke shines this really powerful blue light.
And blue light is, unfortunately, a complete non-starter for looking through human tissue.
Why is that?
Well, you have to remember what our tissue is made of, right? Mostly water, muscle, and blood. Blue light hits those things, especially the hemoglobin in our blood and the water.
And it just gets eaten up.
It gets absorbed incredibly efficiently. It's like trying to shine a cheap flashlight through a bucket of mud.
The light just scatters and disappears.
It's devoured. The body is just packed with these things called chromophores that love to absorb blue and green wavelengths. That light never, ever makes it out to a detector on the skin.
So the disease could be shining like a supernova deep in your abdomen, and from the outside you'd see nothing.
Absolutely nothing.
What's fascinating here is that the search for a solution is completely driven by that one physics problem.
It is.
Scientists aren't just looking for brighter light. They're hunting for proteins that shine red, or even better, near infrared.
That is the crucial pivot. Those longer, lower energy wavelengths, the deep reds in the near infrared spectrum, they can penetrate tissue so much better.
Why is that?
Because there's minimal absorption by those main culprits, like water and hemoglobin. That NIR window, as we call it, is really the only way you're going to get a clear signal from deep inside the body.
So the shopping list is clear now. We need a system that glows red, or NIR, doesn't need constant fuel, and can stay safe in the body for years.
And that realization is what's changing the whole philosophy. I mean, we've basically hit a wall with what we can do with these synthetic luciferase systems. The next breakthrough probably isn't coming from a chemistry lab.
It's coming from borrowing blueprints from nature's own engineers.
Exactly.
We should probably pause here and clarify what we mean by these light systems in nature, because the difference between bioluminescence and fluorescence is it's critical for what comes next.
That's a great point. So when we talk about true bioluminescence, we mean an organism that produces its own light, chemically.
Like nanoluke.
Just like nanoluke. They have the enzyme, they have the fuel, they generate the photons themselves.
Which we see in things like jellyfish, right?
Yeah.
They have these luciferase-like systems, and all those deep-sea creatures, fish, shrimp, bacteria, they're all turning their own lights on or off.
Now, fluorescence is different. A fluorescent protein doesn't make light. It absorbs light from an outside source.
Like a flashlight or the sun.
Right, and then it immediately re-emits that energy at a longer wavelength. That's the color we see.
And here's the key biological fact. Mammals, so dolphins, humans, we are not bioluminescent. Our cells don't have these systems. We don't make our own light.
Correct. We do have some proteins that can fluoresce, but they're not the kind of stable, red-glowing proteins we need for deep tissue tracking, which is why so much research has gone into engineering new ones.
Okay, so if that's the case, if mammals don't make light, why on earth are researchers looking at dolphins? A non-glowing mammal? It seems counterintuitive. Why not just double down on the super bright jellyfish we know can glow?
It's a great question. And it really gets to the heart of this deep dive. The key isn't the glow itself. It's the incredible stability and the spectral properties of their proteins. We don't care that the dolphin can't turn a light on. We care about how unbelievably robust its biological hardware is.
Here's where it gets really interesting. Scientists figured out that even though dolphins don't glow, they have these natural fluorescent proteins with exactly the traits we need for long-term tracking.
Yeah.
The traits that nanoluke just couldn't deliver.
Yes, there are gold mines. Because evolution already solved the problems that we were struggling with in the lab. You think about the list we made. We need stability, the right color, and safety.
Let's start with color. How does a non-glowing mammal give us the color we need?
So the dolphin proteins, or at least certain engineered versions of them, they fluoresce in the red and near-infrared regions. That's their signature.
So right away, the blue light problem is solved.
Immediately. These are the wavelengths that can get through tissue. So you can shine a harmless, low-energy external NIR light on the body, and the dolphin protein will glow back with an even deeper red signal that you can actually read right through skin and muscle.
That takes care of the physics. But the real breakthrough, the one that solves the fuel problem from nanoluke, that comes down to the environment these proteins evolved in.
Absolutely. Think about it. We're talking about 50 million years of evolution in a really harsh environment.
The ocean?
The ocean. These proteins evolved to be incredibly stable, non-toxic, and resistant to breaking down in a salty, high-pressure, oxidizing world. They're tough enough to last the entire lifespan of the mammal.
A lifetime of stability versus minutes of light from nanoluke.
And that means we get rid of the need for external chemical fuel entirely.
That's the shortcut.
It's the ultimate engineering shortcut. If we can borrow the genetic code for a protein that's just inherently super stable and long-lasting, we don't need to keep feeding it.
You just need to engineer the patient's own cells to produce this stable, dolphin-derived protein.
And then for the scan, you just apply a simple non-invasive light source from the outside to sort of charge up the fluorescence.
So studying dolphins isn't about making us glow in the dark. It's about taking their 50 million year head start on protein engineering, on toughness and color shifting, and using that architecture to build our tracker.
It moves us past the whole luciferase dead end and toward these super stable fluorophores. The goal is pretty clear now. Use the dolphin blueprint to build the next generation of glow tags that are safe, shine red, and can last for years inside a patient completely harmlessly.
And if we connect that back to the bigger picture, this is a real paradigm shift. This leap could give us what we talked about at the start, a real-time, non-invasive, glowing map of disease.
It completely changes how we talk about diagnostics. Instead of one big ionizing X-ray or a long, expensive MRI, you're talking about longitudinal monitoring. Continuous.
So you could get a single dose of these tracking cells, and then for years after, just use simple light scans to see if a tumor is growing or shrinking or coming back.
Which means the benefit for the patient is just exponential. You eliminate that trade-off between frequent monitoring and radiation exposure.
Especially for kids, going through cancer treatment.
Absolutely. The scans would just use light, not high-energy waves. This is what precision medicine is really about. Imagine tracking the tiny shifts in an immune response or seeing if a new drug is working live over time.
You're not waiting for a surgical biopsy to confirm if something came back.
The light map just tells you instantly and non-invasively.
So the ultimate outcome, the gravity of this research, is creating the first harmless, lifetime deep tissue tracker.
And that urgency is what pushes this research into these unlikely corners of biology. We know people need these answers now. If we can get this level of stability and this depth of vision, we can intervene sooner, see if treatments are working faster, and, maybe save families years of painful, anxious waiting.
It also says so much about the whole philosophy of scientific discovery, doesn't it? We said the breakthrough came from an unlikely place.
It validates the whole enterprise of asking strange biological questions.
Right.
Science only moves forward when people are willing to look where nobody else is looking. This breakthrough isn't coming from tweaking a synthetic product in a lab. It's coming from a marine biologist or a molecular engineer being willing to look into the dark, forgotten corners of the world, ocean trenches, weird, non-glowing mammals, forgotten biological pathways, and realizing that nature already solved the hardest engineering problems for us.
It's a kind of engineering humility, realizing the best blueprint for a stable, non-toxic diagnostic has been there all along, shining silently in the ocean for 50 million years.
You have to stay curious. You have to search the places science might have just forgotten to look. The fact that the answer to this complex medical imaging problem might come from a non-glowing dolphin is just proof that the most revolutionary ideas are often hidden in plain sight.
So what does this all mean for us? We started with Nanonuc, a brilliant tool, but its blue glow is limited, it needs fuel, it's hard to deliver, and it's fleeting. It's a dead end for human diagnostics.
And we moved from there to the incredible promise of these stable, red, and near-infrared fluorescent proteins, the ones we're finding in non-bioluminescent dolphins. They offer the toughness, the structural integrity, and the perfect color penetration to create these harmless, lifetime trackers.
No more fuel problem, no more color problem.
All is all.
We're moving from these single, expensive, invasive snapshots to something that could be continuous, safe, deep body surveillance, all thanks to a protein that was perfected in the sea.
And this story, I think, it really reinforces a profound idea that the light we need for the next generation of medicine might already be shining. We just hadn't looked at it in the right way yet. It should encourage you, the listener, to really think about the power of curiosity. That sometimes the answer to a huge human health problem is 50 million years in the making, just waiting in the most unexpected species. Keep looking at the strange proteins. You never know what answers they hold.